Hi everyone, my name is Dr.Benjamin Tamilselvan. I am working as an assistant professor at the Department of Zoology, Bishopur College. In this video, I am going to talk about an enzyme called RNA polymerase. So, in humans and as well as in many other organisms, so RNA polymerase is an enzyme. It plays a vital role in all the living organisms, starting from prokaryotes to eukaryotes. In this video, I am going to talk about in eukaryotic RNA polymerase. Generally, RNA polymerase, it mediates transcription of a RNA. That means it converts DNA into RNA. Here, in this process, it does not require a primer, and it transcribes from 5 prime to 3 prime direction. Okay, so the transcription is initiated by many proteins, and that binds to a specific promoter region in the transcription area. Okay, the promoter binds to the DNA, thereby it recruits RNA polymerase to initiate the transcription process. So the transcription process is the main function in translating the DNA into transcribing DNA into RNA. That RNA will be translated into a protein molecule. So that protein will be a functional unit of all the living organisms. So RNA polymerase, it evolutionarily conserved between prokaryotes and eukaryotic organisms, and they have shown many similarities, but there are slight differences in there. So when you compare RNA polymerase with the DNA polymerase plays a different role, whereas RNA polymerase plays a different role. A DNA polymerase is an enzyme, it replicates DNA, that means it copying a double-stranded DNA into multiple copies. Whereas this RNA polymerase, it only transcribes DNA into RNA, which means it converts double-stranded DNA into a single-stranded RNA molecule. What is the purpose of this RNA polymerase as well as the DNA polymerase? That is, it is to copy the entire genome of an organism, that is the DNA polymerase. Whereas RNA polymerase is to copy the RNA from genes. When it occurs, the RNA polymerase, that means the transcription occurs during the G phase, and whereas the DNA polymerase occurs during the S phase. That means replication. Okay. Here RNA polymerase, it does not require any primer. Okay, primer is nothing but a small oligonucleotide that binds to the DNA, and thereby it copies its double-strand DNA. Okay, so here in RNA polymerase, it converts, that means it synthesizes DNA into RNA. While converting DNA into RNA, so you have adenine, guanine, cytosine and uracil in the RNA template, whereas in the DNA template you have adenine, guanine, cytosine and thymine. The thymine here, it gets replaced by uracil when it is transcribing into a RNA molecule. So here in the transcription, the double-stranded DNA will become a single-stranded RNA. That means it produces mRNA molecule. Whereas the DNA polymerase, that is during replication, it copies and produces multiple DNA sequences. So that is the main difference between the function of DNA polymerase as well as RNA polymerase. So if you see in a eukaryotic organism, there are 3 RNA polymerases, whereas in a prokaryotic organism, that means in a single cellular organisms, there is only one RNA polymerase, whereas in a multicellular, like in humans, we have 3 RNA polymerases. All these 3 RNA polymerases, they play a different role, whereas in the prokaryotic organisms, the single RNA polymerase, it carry out all the, or it synthesizes all types of RNA molecules. For example, as you see here, the RNA polymerase in prokaryote, it produces RNA polymerase 1, it synthesizes 5.8S RNA molecule, and then 18S RNA molecule, and then 28S rRNA molecules. So the rRNA is nothing but a ribosomal RNA. the second RNA polymerase, that is the main player, it converts the double strand DNA into a single stranded RNA. So that is the mRNA molecule, the messenger RNA. Okay. Along with the messenger RNA, the RNA polymerase 2, it synthesizes small nuclear RNA molecules and small cytoplasmic RNA molecules. And the third RNA polymerase in eukaryote, it produces 5S rRNA and then tRNA, that is a transfer RNA, and then some small nuclear RNA molecules. All these RNA molecules, they are vital for the transcription process to complete. If any of these RNA polymerases fail to produce one of these components, then the transcription and translation in the eukaryotic organisms will not take place. Okay, and particularly interestingly, this eukaryotic RNA is a monocistronic one, which means it produces only one protein, whereas in prokaryotes, from a single RNA molecule you produce multiple forms of protein molecules, whereas in eukaryotes, from one RNA molecule you produces only one protein molecules. That is why it is called as a monocistronic. In prokaryotic it is called polycistronic. As you can see, so the yellow color one is the RNA molecule. It slides during the transcription process. Here, as you can see here, you have double stranded DNA molecule. Here 3' to 5' strand will be used as a template for producing RNA molecule. As you can see here, from this double stranded DNA, when the RNA polymerase binds to the DNA molecule, and then when it start transcribing the single strand of DNA sequence, it produces single RNA molecule. As you can see here, the RNA polymerase, when binds to the DNA, it starts synthesizing the RNA molecule from this region. Okay, so it uses this as a template, and thereby when it slides along the DNA molecule, it synthesizes the RNA molecule. So this RNA molecule will be further converted into a protein molecule. So that protein molecule is the one is a functional unit. Okay, so here the RNA molecules will be produced by adding nucleotides, that is adenine, guanine, cytosine and uracil. And finally, so this is the transcription and translation in prokaryote and eukaryotic system. As I already mentioned, in prokaryote you produce multiple proteins, whereas in a eukaryote you produce only a single protein. As you can see here, so this is the prokaryotic cell where it does not have a nucleus, it only have cytosol. So the transcription and translation, both the reactions, that means converting DNA into an RNA and then RNA into a protein, everything takes place in the cytosol in prokaryotic system. If you see in a eukaryotic system, the transcription takes place in the nucleus. So once the transcription is finished, the synthesized mRNA molecule further processed and then it is ready to move to the cytoplasm. Once it is entering into the cytoplasm, it produces protein molecule. So that protein molecule will move inside the cell and outside the cell, thereby it regulate many metabolic activities in an organism. So RNA polymerase is a one which is mainly playing a main role to hold all the living organism to function properly for an organism. So with this I would like to conclude. Thank you.